Hello, everyone, and welcome to our third webinar of the series SBBC ISSCR. Before I introduce you today's speaker, we have a few announcements. So the first announcement is a reminder that the SBBC Congress will happen in January 27 to 29 in 2021. Irene, could you please show the slides for the Congress? So the registrations are open and you can register now. You can see there. So you can register for our Congress that is going to happen in January 2021. And so please register and join us for the SBBC Congress. So the second announcement is an invitation from the ISSCR to become a member. So the Society, International Society for Stem Cell Research has a very important activities in the stem cell research field. So please look at the website and become a member. And the third announcement is also from the ISSCR, and it refers to a webinar called Vision 2030. So Vision 2030 gathers business founding scientists and CEOs for in-depth interviews on a decade past and a decade ahead, where we are now, obstacles to overcome, and what is next for our field, business, and careers. This gathering comes at a pivotal moment in regenerative medicine and stem cell science, as discovery, translation, and investment intersect. This digital meeting, this webinar, will take place in January 7 and 8, 2021, so 2021. And registrations will be open soon, and you can check it at the ISSCR webpage. Now I'd like to introduce our speaker, Professor Cédric Blanplain, who I thank very much for being here with us today. It's an honor to have you. Cédric Blanplain graduated as a medical doctor and received his PhD in medical sciences and was a board certified in internal medicine from Université Libre de Bruxelles in Belgium. He was a postdoctoral fellow in the laboratory of Uniflux at the Rockefeller University. And now he's a full professor, a Well Bio Institute investigator, and director of the Laboratory of Stem Cells and Cancer. Cédric Blanplain received prestigious awards, including EMBO Young Investigator Award, the Outstanding Young Investigator Award of the ISSCR in 2012, the Lilian Bettencourt Award in Life Sciences, also in 2012, Joseph Mason Award from FNRS in 2015, and he has been elected member of the EMBO in 2012, the Belgian Royal Academy of Medicine in 2015, and the Academia Europea in 2016. So, Dr. Blanplain, thank you again for accepting our investigation, and the floor is yours.
Thank you very much for the kind introduction. Thank you very much for inviting me to share our result, and thank you all of you for listening. And so today I'm going to discuss with you the mechanism that regulates multipotency in epithelial stem cells. And for that, I will illustrate these with the study in two different tissues, which is the mammary gland and the prostate. So as you know, the stem cell reside on the top of the hierarchy of the cells in the tissue, and they have two essential properties. Their ability to self-renew, so giving rise to themselves, and their ability to differentiate it into one of all the different lineage that compose the adult tissue. In between the stem cell and the differentiated cells, sometimes, but not always, you have a pool of transient amplifying cells or progenitor that will amplify the number of cells that will eventually differentiate it from the division of one stem cells. And the stem cell has the property to differentiate, and different stem cell has the property to differentiate into different lineage. The most primitive, the morula, has the ability to give rise to all the somatic cells of the individual, plus the extraembryonic tissue. And then you have the pluripotent stem cells, which reside in the blastocyst, and that can be cultured transiently or for longer period of time, and then re-inject into the embryo and reform all the lineage, but not the extraembryonic tissue. Then you have multipotent stem cell, and the best example for multipotent stem cell is the hematopoietic stem cells that can differentiate at the clonal level into all the blood lineage. And finally, you have the unipotent stem cell, so a stem cell that has the ability to only differentiate into one of the given lineage. And for example, the best known unipotent stem cell were the spermatogonia, which only give rise to the spermatids and spermatozoid at the end of the day. And so for a long time, it was thought that the adult tissue contained essentially multipotent stem cells. But I'm going to show you some evidence that actually many tissue now, when you look them carefully, they are not multipotent, but actually they are more unipotent, a different pool of unipotent stem cells. So in my lab, we are studying different tissue and organs, and we are particularly focused on the skin epidermis, which I'm not going to talk to you about, the mammary gland and the prostate. And the mammary gland and the prostate, although a priori would be very different tissue, actually are composed of very similar type of cells, the basal cells and the luminal cells. And the luminal cells are the cells that secrete the fluids and the milk for the mammary gland and the seminal fluid for the prostate. And so if you check under the microscope, the cells of the mammary gland is like that. It's different tube. And so in this cross section, you have, the basal cells here and are in the other part of the tube, and the luminal cells here in the inner part of the tube. And in the mouse, the mammary gland developed very early during embryonic development. It actually is the first ectodermal appendage that developed even before the hair follicles, and they develop from the ventral skin here, or the lateral skin of the mouse, as 5 pairs of mammary gland, and they initially form a placode, and this placode will form a mammary bud, and this mammary bud will eventually progress and invade the underlying mesenchyme that is called the mammary fat pad, and very early during embryonic development already develop the first branches. And these branches will, and this different tube will progressively grow during pubertal development to reach and to fill the mammary fat pad, and due to the action of the estrogen. And then during the adult life, you have cycle of growth and degeneration according to the estrous cycle. But when the mouse get pregnant, the mammary gland will further develop to eventually produce milk-producing cells. And as you see here, there's a little bud that branch everywhere, and at the end of the gestation and during the lactation period, the whole mesenchyme is full of milk-producing cells. And then the mammary gland, after the end of the lactation, the mammary gland will involute and more or less reach its virgin adult appearance. until a new cycle of either a stress cycle or pregnancy will occur. And for a long time, people have studied the mammary gland using a transplantation experiment. And so in this experiment, you transplant a piece of mammary gland or even a group of cells or even one single cell sometimes in the mammary fat pad. And so, which is as I told you, the stroma of the mammary gland. And the stroma will help the cells or the piece of the tissue that you transplant there to regrow and to reform a functional mammary gland. And in almost 15 years ago, two groups, the group of Connie and Jane Wiesweder, made a very important discovery, is that the isolation of basal cells here, in contrast to the luminal cells, they can reform a whole mammary gland upon transplantation. Whereas when you transplant the luminal cells alone, nothing happens, they don't engraft and they don't produce the different cell lineage of the mammary gland. And when you stain and you section this mammary gland as it regrow, they present all the different cell types that are present in the mammary gland. And so, obviously, these data were interpreted as the following. There's long-term stem cells that present basal characteristic and which will give rise to luminal progenitor and basal progenitor that will give rise to their differentiated cells. And that's, everybody who had done this experiment would conclude about the same. However, something that we learn in the skin a little bit before the mammary gland is sometimes when you transplant the stem cells, they will present a wounding-like reaction. And during this wounding-like reaction, sometimes the stem cell that you transplant can give rise to cells that normally they don't give rise to. So it was important to assess the fate of the basal and the luminal cells during physiological condition and not during the regenerative condition, which is the transplantation of the mammary gland cells. And so to assess the fate of the cells in their physiological condition, the best technique to do that is what we call lineage tracing analysis. And so what is a lineage tracing analysis? It's you use lineage-specific promoters, in this case either a basal promoter or a luminal promoter, that express an inducible Cree, and the inducible Cree can be, for example, activate here if it's a Cree ER by the tamoxifen administration. And upon the activation of the Cree, they will remove a stop cassette that prevent the expression of a fluorescent reporter protein, and these fluorescent reporter protein are expressed then in the cells that you target and all their progeny. And so we generate to that new Cree line that were targeting specifically the luminal cells or targeting specifically the basal cells. And the aim of this experiment, it was to determine in which type these cells that we trace can differentiate into. And so either we trace a multipotent stem cell and then it will give rise to different lineage, in this case, the basal cells or the luminal cells. And if we trace a unipotent stem cells, the cells that we trace will only give rise to one of the lineage that compose this tissue. And so we perform lineage tracing analysis just at the beginning of the puberty, just before the pubertal expansion, and we nicely label the myoepithelial cell or the basal cells here, as you see, and no inner cells are labeled initially. But when we look at the end of the pubertal development, even after the cycle of lactation and pregnancy, we were very puzzled by the fact that the basal cells that we lineage trace here only give rise to basal cells and not the luminal cells that should appear in this gate. And here's the quantification. You see, here we label 50% of the cells, and since the tissue had expanded considerably, if this basal cell should have given some luminal cells, but that did not occur. So we asked the question: would it be possible that the luminal cell lineage actually act independently of the basal lineage and are able to sustain and produce the luminal cells during this period of pubertal expansion? So when we trace the luminal cells here, we specifically trace only luminal cells and not basal. And 10 weeks after, we were very intrigued by the fact that indeed the luminal cells only give rise to luminal cells, but their proportion didn't decrease. Or if they were replaced by another cell type or multipotent cell type, they would dilute the label cells, and then the chimerism of the label cells would eventually decrease, which did not happen. So that suggests that during this physiological condition, and including the pregnancy and the lactation, the luminal cells and the basal cells are maintained by independent cell lineage and independent stem cell that maintain this lineage. So, but then why people has found that the basal stem cell are able to give rise to all the cell type in transplantation? So we use our lineage tracing strategy to lineage trace the basal cells and then isolate them and graft them into the most mammary fat pad. And in that circumstance, as you can see here, we can also reform a complete mammary gland upon transplantation, and the cells that we transplant can give rise to the luminal and the basal cells. So the same cells that are unipotent during physiological condition become multipotent in transplantation. And the question was, what are the mechanisms that maintain this unipotency during physiological condition, and why this mechanism is disrupted during transplantation? And at the end of my talk, I will give you an answer to that particular question. So, what I show you here is in adult mice or in postnatal mice, there's two independent lineage restricted stem cell that maintain the luminal lineage and the basal lineage. And the question was, is at the beginning of the embryonic development, is there some multipotent progenitor that give rise to this two lineage, or already the lineage restriction had occurred at the early stage of embryonic development? So that's what we did, and we then induce here, we perform lineage tracing during this early stage of embryonic development. And for that, we use a dox-inducible system in which we can give a very, very tiny dose of doxycycline in a way to label one and a single cells in the embryonic mammary bud at E13. And then we analyze the fate of the cell that we label in postnatal mice, during which the basal cell and the luminal cells are already segregated. And indeed, when you do this experiment, you realize that when you label one cell in the mammary gland, these cells can give rise to more than one lineage, both basal and luminal cells at the same time, and were indeed bipotent during this early stage of embryonic development. However, as soon as the mice are born, and when we do the basal lineage tracing at P1, then all the cells the basal cells are already lineage restricted and exclusively give rise to the basal cells. So then the question was, okay, what is distinguishing the multipotent cells from a unipotent basal or luminal cells? And to do that, we perform transcriptional profiling and RNA sequencing, and we isolate the embryonic mammary bud at E14 when they are all multipotent, isolate them by flow cytometry, and perform single-cell RNA sequencing. And as you see here, this embryonic progenitor, they segregate differently than adult basal cells and luminal cells, and they are a little bit more closer to the adult basal cells than the luminal cells. And as you see here, the luminal cells come into two flavor. I will come back to that in a minute. And so then we ask the question, is this embryonic progenitor, are they co-expressing marker of basal and luminal cells at the single cell level? And as you can see here, when we plot the proportion of luminal cell marker or the proportion of basal cell marker, whereas the basal cell are, strictly basal cell and do not express luminal cell marker, this embryonic progenitor, they are hybrid and they co-express basal and luminal cell marker at the single cell level. And so what defines really the multipotency at this early stage of development is this dual transcriptional program that drive the co-expression of two different lineages. And then after birth, these two different programs will eventually resolve into one luminal differentiation program or one basal differentiation program in the different cell type that will compose the postnatal mammary gland development. So we published this story 2 years ago together with the laboratory of Sylvia Frey, who really found very similar findings. And the only difference and addition to in the works of Sylvia, she could show that already the lineage restriction was occurring at the late stage of embryonic development around E17.5, and confirming what we found in our postnatal P1 lineage tracing, that both cell types were segregated. So as I show you in my single cell data, the luminal cells come into two groups of cells, and so it's known for a long time that if you stain the luminal cells here in magenta, you see that some cells here in red are expressing estrogen receptor or progesterone receptor, so the receptor for the sex hormone, and it's more or less half of the cells express estrogen receptor, so ER or PR, and half of them do not express this marker. And the question was then, is this different, two different compartments within the luminal cellular compartment, are they maintained by one single population of stem cells, or are they also themselves self-sustained by their own pool of progenitor? And so that's another image showing you that the PA expression in the mammary gland. So to do that, we generate a new lineage tracing strategy that allows us to activate the YFP expression only in ER positive cells. Oops, sorry, and not in ER negative cells. And you see, in the absence of DOX, this CRE is not leaky, and upon DOX expression, the YFP is expressed only in the cells that are here ER positive. And you see here, 4 weeks after, all ER positive cells are labeled and not ER negative cells. So this means that during the period of postnatal expansion and pubertal expansion, the luminal cells were segregated in two pools of cells, and that are sustained by their own pool of progenitor, and the ER positive cells sustain the ER positive lineage, and as you see here, by the stability of the chimerism of this different cell type. During pregnancy, there's a massive expansion of the ER negative population, which is the milk-producing cells. And there's even a progressive loss of ER expression in the rare YFP positive cells, the ER positive derived cells that remain. And during lactation, as you see here, there's no more expression of the ER. But interestingly, after the involution, when all cells, the luminal cells are replaced, and there's a new equilibrium of ER plus and ER negative, and all the ER positive cells, they arise from the cells that were initially ER positive cells. So even though they lost the ER expression, they maintained the memory that they were an ER positive cells, and they replenish themselves at the end of the involution. And you see here the chimerism. Although it's been diluted dramatically during pregnancy, this chimerism go back to its normal level after the involution and even after several cycles of involution. So that really demonstrates that the ER positive cells are not terminally differentiated cells, as it was initially suggested, but they contain ER positive stem cells that sustain their own lineage. And two different groups that published this paper about the same time than us found that the ER negative cells also contain their pool of long-term stem cells, and that can be labeled here by Notch 1 as the group of Sylvia Frey, I've shown, and the group of Guo has used different mice, but also consistent with what I told you here, that the luminal cell compartment is a heterogeneous compartment, both of them sustained by their own pool of progenitors. So in contrast to what we were thinking, that initially that the mammary gland only contained multipotent stem cell and all the rest are either transient amplifying progenitor or terminally differentiated cells. Actually, all the different lineage of the mammary gland in the postnatal mice are sustained by their own pool of unipotent progenitor. So that was a very big shift in the way we conceive the multipotency in the mammary gland. So here I'm going to show you some data that has been gathered by Maria Lucet, former postdoc in my lab, and Elsa Tica, a very talented postdoc, PhD student. And what they did is they performed clonal analysis in the prostate. And so, as I told you, the prostate, although a priori it's very different architecture than the mammary gland, when you look at the cells that compose the prostate, it's very similar. You have luminal cells that are surrounded by basal cells here, and once in a while you have neuroendocrine cells that are quite rare. Cells that are intertrichate in between basal cells. And so when Marielle performed the lineage tracing of the basal cells during postnatal development, you label initially here, the basal cells, but, few weeks after, these basal cells has given and differentiate into the luminal cells. Here you can see that by quantification, initially you label 30% of the basal cells, but then 4 weeks after, you have a massive expansion of the luminal cells that are labeled by this fraction of the basal cells. And also, we could show that even the neuroendocrine cells here, expressing this marker here, can also arise from the differentiated of a basal cell. So, in contrast to the mammary gland, in which all postnatal development is mediated by unipotent cells, in the prostate, at least at birth, there's still the basal cells are still multipotent. So what Elsa Tica did is to perform lineage tracing at different time during postnatal development. So when she performed here lineage tracing at birth, you see here all the cells, all the basal cells give rise to the luminal cells. in the duct region or in the tip region, so irrespective of the location. But when she did this lineage tracing at 10 days, then the situation was very different. All the ducts were only, the basal cells only give rise to the basal cells and no luminal cells anymore. And however, at the tips region, where at the end of the tube, then they were still, multipotent stem cell, since these basal cells can give rise to the differentiated of the luminal lineage. But as soon as 3 weeks postnatally in the prostate, when you trace the basal cells, they only give rise to basal cells, and as you see here, no luminal cells are labeled in this tissue, and so suggesting that the prostate also undergoes a switch from basal cell multipotency to unipotent basal and luminal cells, but this switch do not occur exactly at the same time, and the mammary gland, it occurs very early, whereas in the prostate, it occurs sequentially, first along the duct region and then at the tips region, to end up by being unipotent at 3 weeks postnatally. And then in the prostate, as in the mammary gland, all the pubertal expansion are mediated by unipotent stem cells. When you do in the adult prostate, you can castrate it, this mice, and add back androgen. And so during this cycle of castration regeneration, when you put the androgen, the prostate re-increase in size, we check whether there was unipotent stem cell or multipotent stem cell that sustain the prostate regeneration. And here, as you can see, all the regeneration of the basal cells here had occurred through only the basal cells, whereas all the expansion in the luminal lineage arise from the luminal cells, and there were no multipotent stem cell found. And that finding is consistent with the finding of different group, in particular the group in Baylor. So what I show you here is for the prostatal development are mediated by bipotent basal stem cells, whereas in postnatal development and adult maintenance, castration and regeneration, they are maintained by the distinct pool of stem cells. So now I will end by discussing with you, what could be the mechanism that restricted the multipotency in basal stem cell during homeostatic condition. Because I told you, these cells, they are fully capable of giving rise to all the cell lineage as soon as you transplant them. You take them out of the tissue, you transplant them, they immediately activate this multipotency. And so when we co-transplant although basal cells and luminal cells at the same time, then the basal cells remain unipotent. And so we hypothesize that the basal stem cells, the luminal cells, and the basal cells, they undergo a crosstalk in which the luminal cells tells the basal cells, we are there, we don't need you basal cells to sustain our own lineage. And so to assess whether there was a communication between the basal cells and the luminal cells to restrict basal cells multipotency, we generate a genetic system in which we can lineage ablated luminal cells, and at the same time lineage trace here in red all the basal cells. And when we, by giving rise to doxycycline, we kill the basal cells and see whether then the basal cells can give rise to the luminal cells. And it's exactly what we have done here. That's the mammary gland in the absence of doxycycline. As you see, the red cells are only in the K14 lineage and not in the luminal cells. And as soon as you give rise to dox, there's a massive flux of basal cells into the luminal cells to repair the mammary gland. And the same was also true in the prostate. When we give drugs to the prostate, the basal cells, although in this adult mice normally they only give rise to basal cells, being they are activated and they produce new luminal cells. We also did that in different other different tissues, such as the salivary gland, the sweat gland, all glandular epithelia composed by basal and luminal cells that were previously shown to be self-sustained by their own pool of unipotent stem cell and progenitor. So what I show you is kind of a generic phenomena across the glandular epithelia, that the multipotency is restricted by a signal sent by the luminal cells to the basal cells and restricting their multipotency. So the question was, okay, is this signal occur through the recruitment of inflammatory cells, which indeed can happen, and in this circumstance, and so we turn out to get rid of the stromal cells, and we turn out to organoid cells in vitro without stromal cells, and in this condition, also the lineage ablation of the luminal cell can also massively promote the differentiation of the basal cells into the luminal lineage in vitro in the boundary of the stroma. So what this experiment means that the mechanism that restricted multipotency in the mammary gland are cell intrinsic mechanisms that are independent of the microenvironment and the stromal cells, or the activation of the immune cells following cell death. So to understand the mechanism that mediated multipotency upon lineage ablation, we also perform a single cell transcriptomic here, and what you show here is the basal cells. That's the basal cells in the wild type situation. And this little blue cells here is the basal cells that are hybrid in between basal and the luminal cells. That's the two population of luminal cells, ER positive and ER negative, that I already talked to you about. And as you see here, these basal cells, they form a continuum in between this different lineage and eventually differentiate into. And when you check, like we did during embryonic development, whether these hybrid cells, they also co-express basal and luminal markers, the answer is yes. They massively co-express at the single cell level both transcriptional program of the basal and the luminal lineage, and so probably multipotency in these two conditions, embryonic development and lineage ablation, are mediated by the same mechanism, same molecular mechanism that goes through the transition in which the two transcriptional programs of the two cell types are co-expressed in the single cells in the activated stem cell, which will eventually resolve and differentiate into the ER plus or ER minus lineage. So to try to define what could be the molecule that the luminal cells secreted and are activating the basal cells to restrict their multipotency, we use a software that's called cell phone DB. It's a software that has been developed in the laboratory of Sarah Teichmann in Cambridge. And what a laboratory can they put all the ligand pair interaction, and so with your single cells, you can compute the probability of having an interaction of a ligand pair interaction, and more the probability is bigger are this little board here, and the mean expression can be also seen here in the color. And so we only check what are the ligand pair interactions that are predicted to function in the absence of ablation and in the absence of in the adult life and not in the embryonic progenitors. And we found several pairs: TNF, TNFR1, PDGFRA, BMP activin receptor, and beta-1 integrin and collagen. And so we try to inhibit this pathway, and here by giving rise to an anti-TNF antibody, a PDGFR inhibitor, an activin receptor inhibitor, or a beta-1 blocking antibody. And when we did that, only one of this condition block the interaction between TNF and TNF receptor, promote the activation of multipotency in the organoid in the absence of luminal cells ablation. And in contrast, and so that's also, it works also both in organoid in vitro, but also in vivo when we administer this antibody to the mice. The basal cells can give rise to the luminal cells in the absence of injury. When, in contrast, when we add TNF alpha and we lineage ablated the luminal cells, which normally would activate the basal cells and give rise to luminal cells, then the TNF alpha signaling inhibit this activation of multipotency and only give rise to the basal cells. So clearly, TNF alpha was an essential mediator of restricting the basal cells multipotency during physiological conditions. I don't mean by that other molecules cannot do this job, but certainly TNF alpha can do it. Then we checked, what would be the signal that are activated in the basal cells following lineage ablation, and we found that 3 types of signaling pathway, Notch signaling pathway, Wnt signaling pathway, and EGFR signaling pathway are activated following the luminal cell ablation. And when we block either the Notch pathway, we block the activation of multipotency. When we block Wnt pathway, we also block the activation of the multipotency. And when we block the EGFR pathway, RBB pathway, we also majorly impair the activation of multipotency. So we found here that, the TNF alpha was restricting multipotency, whereas Notch, Wnt, and EGFR are promoting multipotency upon damage. And now what we are trying to understand is, what are the common pathways that activate multipotency in all the different conditions, in embryonic development, upon lineage ablation, upon transplantation, which I didn't talk to you about, and about oncogene expression, which also I didn't talk about. And indeed, when you cross all the signature in which multipotency was activated in this different cell, we indeed found some very common signaling pathway, and now we are trying to investigate their function and check how they are acting to promote or restricted multipotency in all these different conditions. So with that, I will thank all the people who did this work. Alessandra Van Kemelen was the person in my lab who initiated all the mammary gland studying in my lab. Marco Fioramonti, postdoc, Shuangning, postdoc that work on the lineage ablation project. Alessia Sentonze, a PhD student who was the first author with Shuangning on the last lineage ablation paper. Elsa Tika, who work on the prostate and who also contribute to the lineage ablation paper, and Gaël Bouvancourt, who provide us very good technical helps. And with that, I thank our collaborators, Ben Simon and Stefan Rollins, for the mathematical modeling, and Thierry Vout and Alessandro Zifrim for the single cell transcriptomic analysis. And that, I thank all the rest of my lab, which is a wonderful group of people, and our very generous funders that allow us to work on this different project. And with that, I thank you for your attention. I will be very happy to answer your question.
Thank you, Cédric. There are several questions from people watching from in the YouTube. So I think we can start. Yes, the first one by Irene. Does the multipotency in basal cells vary with the estrous cycle or age of the donor animal?
No, so for the moment we have tested during physiological condition, including the estrous cycle, including during pregnancy, including during evolution, and we could, even in 3 pregnancies in a row, we never see basal cells that activate their multipotency. So you need really to have a disruption of the normal program to see multipotency to occur. However, the stemness and the clonogenicity that can indeed vary with the age and with the potential with the estrous cycle. That's something, it's an excellent question that is not well studied. And so Xuan Lin in my lab are trying to tackle this different question and check, really. But as you may know, the estrous cycle in the mouse is very, very rapid. It's a few days. And so it's always challenging to capture the cells in the right estrous cycle, but it's something that we still take the challenge ahead and we investigate this current question.
Thank you. So the next question, let's see which one it is. By Natalie, is the progesterone receptor expressed by the ER positive cells during pregnancy?
Yes, exactly. And so that's a fabulous question. Yes, and indeed, this is the mechanism that we think are responsible for this memory. I show you that eventually the ER positive cells lost ER expression, but they still maintain PR expression in these cells. And so that probably it's what reinitiated their identity and the expansion following the involution. Yes.
Okay. Thank you. The next one by Victoria. Do you know the evolution of the genetic signature of key genes between the multipotent embryonic precursor and the unipotent postnatal lineage?
Yes. So, it's still, not completely clear. So what we are doing now is, and the group of Jeff Walsh already kind of tackled this question by doing ATAC-seq at the bulk level, and really provide, because with the ATAC-seq, so allowing you to map the open and closed chromatin region, you can infer, if you want, what are the transcription regulation that occur at that particular enhancer. And so you can, kind of gather the gene regulatory network there. But this has not been done yet at the single cell level, and no, probably the single cell ATAC-seq and combined with ChIP-seq would be a very good way to further define this epigenetic landscape of the embryonic progenitor and the unipotent stem cell. But still a lot of things to do there, and it's incompletely understood clearly.
Okay, thank you. The next question. What happens with the ablation of basal cells in the mammary gland and prostate?
Yeah, so that's an excellent question as well, because we could show that with the oncogene expression, the luminal cells are quite good at giving rise back to the basal cells. So we try doing the same kind of lineage ablation of the basal cells and lineage trace the luminal cells. And at least in the condition that we tested, the basal cell, the luminal cell could not give rise to the basal cell. However, this is something important. In the trachea, there's a group of in Boston who show that if you lineage ablate all basal stem cells, then the luminal cells can revert back. But you need to lineage ablate more than 99% of these cells, which we didn't, achieve at all in our experiment. If we, because as you may know, the keratin 14 that we use to lineage ablate the basal stem cell, they're expressed in the skin itself. So if we ablate everything, it's the whole mouse that fall apart. And so and if we lineage ablate, let's say, a fraction of those, then these luminal cells cannot give rise because probably there's still too many basal cells that are there. And so I think, the failure, but, it's just thinking, or failure may, due to the fact that we cannot lineage ablate most, if not all, of the basal cells. So the jury is still out to whether or not in this condition the luminal cells can do the job.
Okay, thank you. The next one, is it known how progesterone receptor help to regrowth the ER-positive cells after pregnancy?
No, that indeed, I'm not sure that we have profiled these cells during this period of time. Certainly the progesterone signal and activate the transcriptional program, but which are the group of genes that they activated to reform and regenerate the pool of ER-positive cells, I don't know that. And I think also it's a very important question, and I will make sure that we should profile these cells at this different stage to address this question, because it's a very good one.
Okay. So, okay, and so Natalie also asks if you look at the pathways when the basal cells were transplanted into.
Yes, we did that. So that's the work that we are working on for the moment. So we transcriptionally profile the basal cells when they were transplanted alone, and we profile the basal cells when they were transplanted together with the luminal cells, to see again what in the presence of the luminal cell, what are restricted, and in the absence, what are activated. And so that we indeed did that, and we compare this pathway to the one that we found in lineage ablation and the one that we found in oncogene expression. And now we are trying to functionally validate the different candidates that we found in common across all these conditions to see what could be a common mechanism, a universal mechanism that regulates multipotency there.
Okay.
But all excellent question from Natalie.
So I have just one final question from myself. I was wondering if this early lineage segregation happens in other epithelial cells apart from glandular epithelium.
Apart from glandular epithelium.
Yeah.
For example, in the skin, initially also everything rise from relatively common progenitor, but also like in the mammary gland, in the adult, the different lineage, they are also lineage restricted. In the skin, you have the interfollicular epidermis, which make the skin barrier maintained by their own pool of cells. The infundibulum, so the junctional zone, are maintained by their own pool of cells, and all the hair follicle are maintained by their own pool of cells. And when you do a wounding, then all the cells, contribute to the wound regeneration and the wound closure. So also there's also lineage restriction there. In the lung, it seems that the different lineage can be also sustained by their own pool of progenitor, depending on the region of the lung. The trachea, that there's basal stem cell all along during adult life, but in more distal epithelia, there's also some cells that are giving rise to themselves and sustain. Yes, and something that also it's intriguing. It was the hematopoietic cells were initially used as a paradigm to the fact that these cells are multipotent and all the replenishment, the daily replacement are mediated by the multipotent stem cells. But actually, it's not the case neither there. And that's a group of Fernando Camargo who did that, with the very elegant transposon-based lineage tracing, that even in the hematopoietic stem cells, the different blood cells are sustained by their own pool of progenitor that are very slowly replenished by the multipotent stem cell. But the daily basis, the daily replacement are also kind of lineage restricted. So somehow from a paradigm that all adult tissue are sustained by their multipotent stem cell, actually we are more and more in a paradigm in which in most adult tissue, the different lineage are sustained by their own pool of progenitor, in which the multipotent stem cell can act as a safeguard there in case of problem or in the long term to slowly replenish the pool of the other cells.
Okay, Thank you. So I think we have just one more question, also by Natalie. I think she was referring to your last answer.
Yes.
But this also means that inflammation has nothing to do with basal cell multipotency.
Yeah, it's always difficult to be very categorical and say, no, inflammation. Inflammation occurs when you do lineage ablation. And when we give rise to glucocorticoid, in which we damp a lot the inflammation, it doesn't change anything to the multipotency. And as I said, it can also happen in vitro independently of the inflammatory cells. But can I really exclude that inflammation do nothing? No, I cannot exclude that, and I think we should. It's possible that they do something, but it's not obligatory. It's not necessary. It's not obligatory.
Okay, so thank you so much. And Jacqueline is saying fabulous data. Thanks for sharing this. And this is we also thank you so much for this wonderful talk and for taking time and being here with us.
Thank you very much for the extremely interesting question and a very good audience. Thank you so much.
Thank you. We hope to have you here someday in person.
Yeah, I hope to. I would love to come to Brazil the second time.
Great. Thank you.
Bye.
Bye.
Bye.